Part 1. Functions and sequences. Key concepts. Function representations. Functions can be represented in multiple ways: verbal, descriptions using language; algebraic, written as equations; graphical, drawn as curves or lines on a coordinate plane; and numerical, organized in tables of input-output values. Each representation provides unique insights. For example, graphs are useful for visualizing trends and identifying patterns, while algebraic forms are better suited for making precise calculations and predictions. In biological sciences, it is important to move fluidly between these forms. The choice of representation often depends on the source of the data, for example, lab measurements versus theoretical models, and the goal of the analysis, for example, prediction, interpretation, communication. Being able to convert between these formats helps verify that a model is consistent and enables a deeper understanding of how the function behaves in a real-world biological context. For instance, bacterial growth may first appear in a data table, then be modeled with an equation, and finally visualized as a curve. Types of functions. Linear functions model processes that occur at a constant rate, such as steady drug absorption over time. Polynomial functions are used for more complex biological processes that change direction or exhibit multiple peaks and valleys, such as changes in hormone levels or nutrient concentrations. Exponential functions describe rapid increases or decreases in quantity. These are commonly used to model unchecked cell division, population explosions, or the decay of substances, like medications or radioactive tracers. Logarithmic functions are the inverse of exponential functions. They are often used when interpreting biological data across wide scales, such as pH levels or DNA sequence similarity scores. Rational functions, ratios of two expressions, and trigonometric functions, based on periodic cycles, also have biological uses. For example, enzyme kinetics often follow rational patterns, while circadian rhythms are well described by trigonometric models. Transformations. Understanding how functions change when manipulated is critical for modeling real-world processes. These manipulations are called transformations. Horizontal shifts, moving left or right, may represent delays or early starts in a biological process. Vertical shifts, moving up or down, can indicate changes in baseline levels, such as increased hormone levels after treatment. Reflections across axes can model reversed or opposite biological responses, such as a feedback loop that inhibits growth. Vertical stretches or compressions alter the scale of the graph, which can help match models to actual data ranges. Exponential growth and decay. Exponential growth occurs when a quantity increases by the same proportion in each time period. A classic biological example is bacterial reproduction, where each generation multiplies the population. Exponential decay models situations where a quantity decreases over time, such as the clearance of a virus after treatment or the natural breakdown of a drug in the body. These models are especially useful for analyzing data over time, estimating time to full decay or maximum growth. These models also reveal patterns that are not obvious from raw data, especially when dealing with fast-growing or fast-depleting biological processes. Understanding how to interpret and apply exponential growth and decay is essential in fields like epidemiology, pharmacology, and virology. Logarithms and plot interpretation. When dealing with exponential trends, it is often helpful to use special types of plots. Semi-log plots use a logarithmic scale for the vertical axis. These plots straighten out exponential curves, making trends easier to compare and interpret. Log-log plots use logarithmic scales on both axes and are useful when both variables cover a wide range of values. These are common in studying relationships like gene expression levels, or organism size versus metabolism, allometry. Choosing the correct logarithmic base, such as base 10 or the natural base E used in biology, is essential for accurate analysis. Misapplying logarithmic rules can lead to serious errors in interpreting biological data. Sequences and difference equations. Sequences are lists of values at specific intervals, often used in biological models that work in discrete steps, such as tracking the number of infected individuals each day or measuring drug levels after each dose. Difference equations describe how each term in a sequence relates to the previous one. These are useful for modeling feedback systems, disease spread, or recurring biological behaviors. A key point: every sequence or recursive process requires an initial condition, a clearly defined starting point. Without this, the model cannot generate accurate future values. In biology, this starting point might be the initial number of cells, viruses, or the dosage level. These models are commonly used in ecology, epidemiology, and systems biology. Biological applications. Malarial parasite growth. Malarial parasites reproduce rapidly within red blood cells. Their population tends to increase exponentially during early infection. Modeling this process helps researchers understand the urgency of treatment and assess transmission risks. A simple exponential model can predict how the number of parasites changes with each replication cycle. Visualizing the process as a graph can demonstrate how untreated infections grow rapidly and how growth slows or reverses when treatment begins. This example shows how mathematical models can be applied to urgent biomedical problems. HIV decay and treatment modeling. After successful treatment begins, HIV RNA levels in the blood decrease rapidly. This drop follows an exponential decay pattern. By modeling the rate of decay, clinicians can evaluate how effective a drug regimen is and determine the timing of follow-up testing or treatment changes. These models also reveal that low levels of the virus may still exist even when standard tests show no detectable virus, a critical insight for long-term care planning. The mathematical model allows the extraction of time constants, which help quantify the rate of viral decay. These insights support better planning for managing patient therapy and monitoring. DNA analysis and logarithms. DNA sequence analysis often involves large data sets. Information theory and logarithmic scoring systems are used to measure similarity and variability in genetic sequences. Using logarithmic transformations compresses large numerical ranges and allows researchers to draw meaningful conclusions from complex data. These tools are foundational in bioinformatics, where interpreting raw genetic information efficiently and accurately is essential. Log scores are used to quantify genetic differences and help researchers identify related genes or mutations. Applying the correct log base and transformation is crucial for ensuring valid comparisons and insights. Modeling drug resistance with recurrence relations. Drug resistance in bacteria or viruses often evolves over multiple generations. As genetic mutations accumulate, a population may become increasingly resistant to treatment. These patterns can be modeled with recurrence relations, which describe how the proportion of resistant cells changes from one generation to the next. By studying these models, researchers can predict how quickly resistance will spread and adjust treatment strategies, such as rotating antibiotics or combining therapies, to slow or prevent resistance. Recurrence relations also reveal thresholds at which antibiotic resistance becomes dominant and treatment becomes ineffective. These mathematical models help guide clinical and public health decision-making in managing infectious diseases. Common mistakes to avoid. Confusing domain and range. Students often confuse the domain, input values, with the range, output values. In biological modeling, clearly defining the domain and range is essential. For instance, time might be measured in hours or days, while the range might represent a population count or drug concentration. Always check whether the domain makes sense in a biological context. For example, quantities like time or population size cannot be negative. Graphs can be used to verify if a relationship is a function by using the vertical line test. Misunderstanding the domain or range can lead to biologically meaningless conclusions. Incorrect use of logarithmic rules. Logarithmic functions follow specific rules for combining or simplifying expressions. Common errors include mixing up rules for products, quotients, and powers. Always simplify expressions inside each log before expanding or applying log rules. Pay attention to whether the problem uses base 10, common in lab data, or the natural base E, common in biology and calculus. Use the same base consistently throughout a problem to avoid incorrect results. These mistakes can significantly impact your interpretation of pH values, RNA data, or growth rates. Forgetting initial conditions and sequences. When working with sequences or recursive functions, you must have a clearly defined starting value, initial condition. Without it, the rest of the model cannot be calculated correctly. In biological systems, this initial value could be the starting number of cells, the amount of drug administered, or the initial infection level. Never assume or skip the starting point, write it down clearly, and ensure it is biologically meaningful. Forgetting the initial condition is like trying to model evolution without identifying the first ancestor.